Okay, Jack goes out with his papers in hand and he finds that there's some crazy acid rain and his papers are going to get destroyed. And he also copied way more than he needed, and so he needs to adjust it before he sends it off to be translated. So this is our strand of DNA, double-stranded DNA. We are going to treat this like we did in the last video, where we're going to produce from this what's called a primary transcript. So from here, if I consider this to be the promoter side and this to be the terminator side, we are going to get a strand of RNA that we call the primary transcript. Okay, I can just get that fixed. Transcript. And it's going to be a piece of RNA that's made from 5' to 3', which means it's going to match the 5' to 3' strand of DNA. And so we're going to have, everywhere there's an A, we're going to put an A, and C, 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 A, but we're going to put a U where we see a T, and then G, G, U, U, A, U, U, 3'. And so this is our primary transcript. Now this primary transcript is not perfect, so we're going to need to do some stuff to it. The first thing that we're going to do is we're going to add what's called a 5' cap. And a 5' cap is a molecule called 7-methylguanosine, and I'm going to just draw it right there. And there's our 7-methylguanosine attached to the 5' base. And the 5' cap has multiple jobs. You can look it up in your textbook on Wikipedia. The main two that I worry about when I talk about it is it acts as a signal for the ribosome, and it also acts as protection. It protects this end of the mRNA. This mRNA, we're going to send it out into the wild, and in the cytoplasm of a cell, it's not as protected as the nucleus of a cell. There are enzymes whose only job is to eat or degrade or break the chemical bonds between these bases, and this protects the 5' end from that degradation. Okay, so that's added. After that, we're going to do something else. We're going to add what's called a poly-A tail in a process called polyadenylation. So here at the 3' end, I'm going to get rid of 3' over there, and I'm going to add a bunch of A bases to it. A. Then we'll put dot, 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 times 200, many A bases. Those A bases go on and on and on, and if you look up this as well, you'll see the same thing, that it has multiple jobs, and it actually has different jobs or different functions in different organisms with different pieces of RNA that we're making. The main jobs I think that your textbook mentions is that it helps in translation, which is the next stage of this process, and it also acts as protection for degradation, which means that these A bases over here protect this end. And what I like to throw in usually when we do this in class is that they act sort of as a kind of timer, like a built-in fuse for a bomb. All of these bases that I drew here in red, these are parts of the RNA that are important. They're going to code for whatever we want to code for. These are pages in the document that Jack made that are important. They're useful pages. Most of them are useful. And then Jack added an extra like 200 blank pages at the end. These 200 blank pages, eventually they are going to be eaten. They're going to be destroyed. They're going to be knocked off one at a time by enzymes that degrade RNA. And slowly we're going to work our way to this point. And then the actual bases here are also going to be degraded. Because this piece of mRNA, we don't want that piece of mRNA to last forever. We want it to eventually be degraded. One of the more important things when we work with complex cells, for the cells to do, is to have very fine control over what's made when it's made, and how much of it we make. And if we have a piece of RNA here that lasts forever, if we had protection at this other end that couldn't be degraded, couldn't be destroyed, then we would be constantly making the product of this mRNA, which we don't necessarily want. We want it for a little bit of time, and then we want to stop making it. If I need more of whatever product I want to make, I can go back to the DNA, get another mRNA out of it, and make a bunch more protein. And then this will eventually be degraded, and I can go back to the DNA, and I can control, or your cell rather, can control how much it makes, when it makes it, and when it stops making it. So our primary transcript has a 5' cap. It has a poly A tail. The next thing we're going to do to our molecule is we're going to do probably the coolest set of words that I ever use in my teaching career. We're going to do what's called intron excision and exon splicing. And this is a fancy way of saying we're going to take some of the pages here that Jack has. We're going to take some of our code of our primary transcript, and we're going to take it out. And we're going to stitch everything together. So, for example, if this piece right here, this AAU, I somehow find out that those pages that Jack made, those pages are not useful to me, somehow your cell knows, your enzymes and molecules within your cell know that there's a section in the RNA that you have that's not useful, that section is going to be removed. So this AAU is going to come out, and this AAU we call an intron, and we are going to excise or cut out that intron, and we're going to splice together the exon or rejoin the exon. So my new molecule is going to have, I'll draw this here, we're going to have our 5' cap right there. So our 5' cap. Then we're going to have our bases, and our bases are A, C, 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 C. A, U are gone. We got rid of them. So now we have G, G, U, U, A, U, U. And then I have, I'm switching colors just so you can keep track, but really all these bases would be the same. A. I'm going to do my dot, dot, dot times approximately 200. Not exactly 200, approximately 200, maybe 250, maybe a little bit less, depending on the cell, depending on the organism. And this molecule here that I've removed the intron, this now with a poly A tail, with a 5' cap, this molecule is called mRNA. This is our ready to go document. We are ready to send this to the translator. We can walk it over there. The translator can look at it, give us all the information that we need. It is protected. It's ready to go. We're ready to get protein from this piece of mRNA. mRNA, for those of you that didn't read ahead, the M stands for messenger. So this is messenger RNA. In the next video, we're going to talk about a bunch of other types of RNA. mRNA is very important, but there are a few others in this process that are important as well. And so these are the post-transcriptional modifications, and the goal of this process, of this set of processes, is to get our primary transcript ready to be translated, to get it out into the cell where it won't be destroyed. It has all the right information that we need, and we're going to do some translation.